here with the respiratory system and starting with the anatomy of that system, looking at an overview. So the function of the respiratory system is gas exchange. It's where gases, namely oxygen and CO2, are transferred across the pulmonary capillary beds, and those gases are then transported throughout the systemic circulation, right? It's going to go through all of our large arteries. And then at the capillary beds, we've talked a lot about the circulatory system, different branches of the vasculature. At the capillary bed specifically is where that oxygen is then dropped off, CO2 is picked up, and then this is transported in the opposite direction for exchange again. So it is the whole purpose of the circulatory system to facilitate the gas exchange that is happening in the respiratory system. In terms of the internal respiration, this is going to support oxidative phosphorylation. All right, going way back to AMP1 when we talked about cellular respiration, the processes, glycolysis, the Krebs cycle, oxidative phosphorylation. This is the whole purpose of oxygen consumption, is to have that oxygen go into the mitochondria and be the final electron acceptor to drive oxidative phosphorylation, which makes us a lot of ATP, right? You can see that process down here. So oxygen's role in terms of why we need it in our tissues, why we need it absorbed into our cells, is to facilitate oxidative phosphorylation, which allows us to make the ATP that drives all of our cellular processes, all of our machinery, transporting hormones, proteins, muscle contraction. All of that is required from the ATP that we make with the help of oxygen. So kind of looking at the end game for where oxygen ends up. The process involves external respiration. So we want to think about all the mechanical components to helping that oxygen move. There's pulmonary ventilation, so the lungs are going to be moving air in and out. There's exchange between the lungs and the blood. So up here at the alveoli, their surface area is lined by these pulmonary capillaries. So the blood and the oxygen are going to come very close in contact to allow that exchange. And then transport happens in the blood, and exchange can happen down here at the capillary tissues. All right. So several different sort of mechanical steps, but the end game is oxidative phosphorylation. Right. So internal respiration is in the cellular phase of this, where oxygen plays its role in oxidative phosphorylation, while external respiration is everything else that precedes that, all the mechanical and transport and exchange that happens before. Okay. Now we can divide the structures of the respiratory system into the upper airway, the respiratory tract, and then there are some accessory thoracic cavity structures like the diaphragm, the ribs, the costal cartilage, right? Those accessory structures help with the movement of air by changing the shape of the thoracic cavity. So we can look at each of these parts separately. Starting with the upper airway, also called the upper respiratory tract. This includes the nasal cavities, right, which are lined by mucosa, the oral cavity, which is another source of taking in air, and then the pharynx. The pharynx is a shared structure. We looked at this in the lab last week. There's the nasopharynx, oropharynx, and then laryngopharynx, and then that leads into the inlet. This is where the respiratory tract itself begins at the opening into the trachea. So everything before the trachea is considered the upper airway. When we talk about things like upper respiratory infections or upper respiratory inflammation, that is going to be anywhere along the nasal cavity, pharynx, and then parts of the larynx. Now we want to think about the functional distinction. So that was more of a structural distinction, upper airway versus respiratory tract. The functional distinction is conducting zone versus respiratory zone. So the conducting zone is where air is simply moving in. There's no exchange happening yet. It's basically a conduit. This is where we're having um, the air being warmed, humidified, any particles or bacteria, pathogens being trapped, but there's not any respiration as such because there's no exchange. And this happens from the nasal cavity all the way down to the respiratory bronchioles. So the conducting zone is going to extend all the way through the trachea, okay, the bronchi, the main bronchi or primary bronchi, the secondary bronchi, the tertiary bronchi, and then at the level of the respiratory bronchioles is where the respiratory zone begins. Okay, so let's kind of look at that here in more detail. So here we have the larynx. Remember, everything proximal to the larynx is the upper airway. But as far as the respiratory tract, it begins at the larynx. However, the conducting zone, which also starts at the nasal cavity, goes all the way down to, excuse me, the terminal bronchioles. If we look at some of the features of the conducting zone, we're going to see that it matches with the function. So we have trachea, which has cartilage. That's going to have lots of C-shaped cartilage rings that eventually get smaller and smaller the further along we go. And then that gets replaced by smooth muscle. So we're going to see as the cartilage decreases, the smooth muscle increases. And these two structures are helping to keep the airway open, right? Helping to keep that tract open and patent, allowing air to flow. So cartilage gradually decreasing, smooth muscle gradually increasing. We're also going to find cilia and goblet cells. Cilia is going to be the apical modifications, right? The hair-like structures that are beating back and forth. This helps with the movement of particles, sweeping particles or pathogens to be expelled, along with goblet cells that are making mucus, and that mucus can trap the particles. So the goblet cells make mucus that traps these particles, and the cilia will simply sweep those particles to be expelled um, in the oral cavity or even into the GI tract. When we get to the respiratory zone, this is where we begin to see exchange. So there's no cartilage, very little smooth muscle, and that is also disappearing. And then also no goblet cells, very little cilia. So at this level, the barrier for diffusion gets smaller and smaller, and at the alveolar sacs themselves, there is no cartilage, no cilia, no goblet cells, no smooth muscle. So all of those accessory structures have disappeared, and here we can actually facilitate exchange because we have the alveoli, which are very thin. We'll talk about those type 1 cells. And then we have the pulmonary capillary, which is on the surface of the alveoli, which is also very thin, and that is perfect to facilitate gas exchange. So these two structures, respiratory bronchioles and the alveoli, are what constitute the respiratory zone, where exchange actually begins to take place. So let's kind of go over that here a little bit in more detail and sort of in a stepwise fashion. So the larynx being the opening into the laryngeal inlet. Epiglottis, we said, is the flap-like covering that covers the glottis, covers that opening. Epiglottis also helps to divert food away from that opening and into the esophagus posteriorly. And then we get into the trachea. The trachea is about 2.5 centimeters wide in terms of its diameter, much thicker and wider than the other branches. It's about 10 centimeters long, and one of its main features are the C-shaped cartilage rings. Now, as we said in the lab, the wings don't go all the way around the trachea. In fact, posteriorly, there's that smooth muscle, the trachealis muscle, that allows for the esophagus, which is behind, to expand as food moves through that location as well. So the trachea will have C-shaped rings which cover the anterior surface only. All right, now we're looking at the bronchi. Now we're looking at the bronchi. These are the main branches from the trachea. So first off, the primary bronchus or primary bronchi, also called the main bronchi. These are slightly smaller in diameter. They have cilia. There's three of these on the right side because we have three right long lobes, while there's only two on the left. That's for the secondary bronchi. Whereas the primary bronchi is only one on each side, right? The right one we said is going to be shorter, more vertical, and wider. The left one is longer, more horizontal, and it's more narrow. As far as the secondary bronchi, we have again three branches on the right, two branches on the left. Here we can see the C-shaped rings are now becoming inconsistent strips or incomplete strips of cartilage, these cartilage plates. So there's still a lot of cartilage, it's just now in these strips instead of very smooth and continuous rings around the anterior. Then the tertiary bronchi are going to be 20 to 23 orders of branching, so very high number of branching in the tertiary bronchioles or bronchi. And then these are going to have also cartilage. The plates here are getting smaller, right? Getting more inconsistent. And then smooth muscle is increasing to sort of take over the role of the keeping the airway open, regulating the diameter of the airway. Then we get to bronchioles. Bronchioles are very, very narrow in terms of their diameter, about 0.5 to 1 millimeter, very, very small in terms of their diameter. And here we see the cartilage completely disappears. So no cartilage at the bronchioles, smooth muscle is increasing, and that is the main structure that keeps the bronchioles open. And then terminal bronchioles is the end of the conducting zone. Here we have no cartilage, lots of smooth muscle, very, very small diameter, and then we have cilia and goblet cells, or no goblet cells, but cilia. Let's look at the overall function of these two zones and some of the different features and components, starting with the conducting zone. So this is, again, just a conduit. We refer to this area as dead space. Air is just sitting in this part of the airway, the conducting zone. There isn't any movement in terms of exchange, and it's about 150 milliliters in terms of the volume that's sitting in that dead space zone. The role of this zone is to warm the air, increasing the temperature to adjust to the body temperature, especially if you're in cold environments or you're in air that's really cold. You want to warm that up and kind of bring that to your body temperature. And that is a part of what happens in this part, in the zone. It also humidifies the air, so adds some moisture, which is important, and then also traps any pathogens. In terms of the epithelium of the conducting zone, it's going to have goblet cells. Here are these goblet cells. These mucus cells are called goblet cells. They help to make mucus. It's also going to have ciliated columnar cells, which are the cells in between. These are the actual epithelia cells. And notice that they have these cilia modifications at their apical surface, which helps with the beating and the movement of any trapped mucus or trapped debris. This phenomena is called the mucus escalator or the mucociliary escalator. So these two things work together. The goblet cells, which make mucus, and the cilia, which can beat or move that trapped mucus with particles. So here's a dust particle, here's a few of them that have been trapped in the mucus made by the goblet cells. And then we can see the cilia sweeping those goblet cell or those mucus globules towards the opening, right, towards the upper respiratory tract. And then a closer view of that, we can see those mucinous granules inside the cells. We can see the epithelia, right, the long columnar cells. Columnar cells are very tall. They have a very long oval nucleus because they have a lot of secretory products, a lot of absorption happening, as well as the energy needed for the beating of that cilia. So very tall columnar cells with these cilia. And then these are also pseudostratified. I think you'll talk about epithelia later on, but these pseudostratified epithelia here means that we have stem cells at the bottom, and then we have these tall columnar cells. So there's kind of this stacking or stratification of the cells of the respiratory tract. So here's a histology view of what this looks like. Deep, we have cartilage. So cartilage is going to be the deepest structure we see here, right? This is like the hyaline cartilage. Then we have a little bit of smooth muscle. Then we have glands, right? Such as those goblet cells. A goblet cell is an example of a gland. It's a unicellular gland. Then we have connective tissue, underlying basement membrane. You can see some arteries here. And then we have our respiratory epithelium, right? Which is at the surface, and then here's the lumen. So at the surface, or the epithelia, we're gonna have our ciliated cells, and then interspersed in between, we'll find those goblet cells.